Assalamu alaikum wa rahmatullah. Welcome back everyone in our second day of research webinar. So today we're going to be discussing the part of any research paper and how to interpret this part and understand it. First of all, the first part in any research paper is the introduction. The introduction is subdivided afterward into several sections. The first one is the background about the, for example, disease or the specific problem we are examining. And the second part is also a background about for example, the diet we are investigating and its impact on the specific disease or the medication we are researching, and etc. Also, we can add the rationale for the study as why there is a need to do such type of research and what they are going to do in this type of research, as we are going to do an interventional trial with certain medication or certain dietary modification. In terms of the results section, usually after the introduction comes the methodology. But in the method, we discussed it yesterday and what to write specifically, including the research site and ethical consideration and etc. After that comes the result. The result can include what we have found, including the figures in specific. And these are, for example, as tables or diagrams, depending, of course, on the method we are using. If we were using any type of tables, then the lower part of any table is crucial to further understanding and the critical appraisal of any specified point in the table itself. Also, the heading of the table, which explains the name of the table and the type of the analysis that was done, is also very important to be included. Moreover, the title can include what they are doing in specific for the diagram and what they are measuring and which is which. For example, this indicates a low fat and this indicates a low carbohydrate, for example. So explaining each drawing or each point in the figure, and also how significant is the value and the time period, for example, in which we have conducted the intervention. After the discussion, after the result comes the discussion, comes the discussion in which we are discussing the importance for this research or type of problem we are investigating, what we have found, and how this implicates the problem we have investigated or any future research. So in the discussion, we can include a result summary, including a snaps of the impact that we have conducted and any other trials that have done the same investigations or similar investigation and what they included. So an integral part of any research is statistical analysis. And in order to have a meaningful result of any research, we must carry out a very successful statistical analysis of the results and the finding in order to be presented in the best way possible and to detect the exact effect. So now we will be discussing how the statistical significance test actually works. So the statistical significance test can aid in the decision-making process in terms of the impact of the problem, as they are mechanisms for helping us to decide whether or not what we see in a trial data can suggest that a true difference exists between two or even more treatment or groups of data. or as a more general definitions that are some effect is actually present in the data. So it is the method that by which we can measure the effect or the effect that is present in our sample. So all statistical significant tests are based on the same logic process of comparing the data between what we actually see with some predictions about the kind of data that we would expect to see if the effect of interest was not present. So this is an example of a biomarker data. These are measurement of the level of a biomarker in patients who were treated with two different medications. So this is divided as all the participants taking treatment 1 and all the participants who are taking treatment B. So calculating the mean value for A is by calculating all the results or all the values in the group A and then dividing them by the total number of participants who are present in that group. So the mean value on A is higher in this example in that of the sample data. But there is a genuine difference in the biomarker levels with these two treatments. And so, if so, how can we decide? So the first step in the logic statistical significant testing is to choose a test static that will measure in some sensible and relevant way the effect in which we are interested in. So in the biomarker example, we are interested in the mean values in the two groups. And so we will choose as a test of statistical significance the differences between the mean values in the two groups. So the value of this test stat is the sample, which is minusing the first group from the second group and having a difference value of 6.9. Therefore, the second step is to establish a sensible null hypothesis for the test. And the null hypothesis involves asking questions about what we would expect the value of the test status that would be if there was no true effect present. And since our test of stat is the differences between the two group means, then under the null hypothesis, this would indicate that we would expect this to have a value of a 0. After that, we can imagine a process of drawing a repeated samples of the data under the null hypothesis, and we can consider the possible values that we might expect to see for our test of stat under this hypothetical processes. So in more details that we can suppose we are able to collect a sample of biomarker of data in circumstances in which the null hypothesis was true. So in that sample, it will consist of individual measurement of biomarker levels in two groups of about 10 patients with no true difference between the group means. So even though that we know if the null hypothesis is true, then the differences between the group means is 0. And in any particular sample, we would not expect to see a value of exactly 0 as the natural sampling variability would probably give us a value that is close to 0, but not exactly as that. For example, as in this picture, it is less than 0 at the point of 0.8, for example, in the minus. And if we suppose that we were able to take a second sample of exactly similar data and under the null hypothesis, this time we would probably see a slightly different result for our observed mean differences. This, for example, would indicate 0.7 in the minus. So if we were able to take a few even more samples of similar data, and for each sample we calculate the differences between the two means, then we would see a range of values ranging from 0 or even up or higher slightly. So this phenomenon is known as the sampling variation and can result from the inherent natural variability of most of the biological and also medical measurement from the patient to patient. And also, it can result in variation within the patient from one time point to the another. And if we were able to take, for example, 1,000 samples of such values, each of the two groups of 10 individual of biomarker measurement, we would get a lot of individual values for the differences between the group means. This is too many to see clearly in a plot of this type, but this is just an example. And we could plot a histogram of the 1,000 mean samples. This will be represented as this, and it can show the frequency with which we get the sample means of a certain value. Therefore, we can think about the process of collecting an extremely large number of such samples of data, and if we are prepared to make a certain assumption about the nature of the underlying individual measurement, then we can find a mathematical curve that will fit the shape of this infinite histogram. So, for example, if we suppose that our individual biomarker measurement have the same normal distribution, as there is no true difference between the group means, and also a common standard deviations, then it can turn out the appropriate mathematical curve to fit the histogram is also a normal distribution with a mean of 0. So with the standard error that is a function of the standard deviation of the individual and also of the sample size. So this can be plotted at a curve such as this curve. So we should know that the mean of this sampling distribution is determined by the mean of the population from which we can draw the sample and its standard error of the spread as a function of the underlying standard deviation and also the sample. In addition, most of the significance tests are based on such mathematical curves, which can represent the sampling distribution of the test stat or the null hypothesis. So we will be focusing now on this mathematical curve for analyzing the research result, as the curve can show the test stat values which might be seen if the null hypothesis is true, and can show the relevant probabilities of getting certain values. We can therefore ask ourselves where on this curve of a possible test stat value, then the result will actually be observed. So a difference between the two group mean of, for example, as the previous example we have mentioned, is 0.9, and this will be located on this point that is red on this curve. So we can think or ask whether or not this value is unusual in relation to the curve that is showing the various possibility. So in other words, in our observed test stat value, is it consistent with this distribution or not? So one way of judging this would be to ask how many possible test of stat values are more extreme than the one that we have actually saw. And we can measure this on the sampling distribution curve by calculating the area of that part of the curve, which is extremely more or even extreme than that of the observed value. So this area that is more extreme is called the p-value, which indicates the probability of seeing a test of stat as extreme or even more extreme than the one we actually saw if the null hypothesis was true. And in this case, the p-value is approximately of 0.15. So how can we interpret this on the drawing as it is shown down there? And this p-value measurement measure the extent to which our observed data are unusual. And this unusual is judged in relation to the mathematical prediction about what other data can have been seen if the null hypothesis was true. And if our observed data are sufficiently unusual, judging this way, then we might be entitled to conclude that our data are inconsistent with the null hypothesis. So conventionally, and it is important to stress that this is just an arbitrary convention, then a p-value of less than 0.05 is taken as an indication, which is the observed data are inconsistent with the null hypothesis. While other p-values could also be used and sometimes are being used, but the convenient value of p less than 0.05 is regarded as the statistically significant value. So this p-value is firmly enriched in the clinical literature trials. So sometimes if this p-value is simply referred to as a statistically significant result, with some researchers saying that when the value is this 0.05, then this indicates rejecting the null hypothesis. But this is not a good terminological presentation, and it is better to say that such p-values will indicate that the data are inconsistent with the null hypothesis as a more of a scientific term. We will be taking a break now for 10 minutes for Al-Asr prayer, and then we'll continue with discussing the statistical method. So one direction can give us what is called a one-sided or one-tailed p-value, while in calculating the p-values by considering as a more extreme values in the both directions, this can give us a two-sided or two-tailed p-value. So whether we use a one-tailed or a two-tailed p-value, the convenience of using a p-value of less than 0.05 as a critical step is still usually being applied. In the two-sided p-values, these are the conventions by which using two-tailed or two-sided p-values is considered a standard practice unless there are any strong scientific argument to the contrary. So according to the guidelines, it is important to clarify whether a one or a two-sided test have been used or will be used in particular to justify the prospectively using of a one-sided test. So how can we interpret the p-value? So we need to know that the first thing is that the p-value is not the probability of the null hypothesis or the probability that there is no genuine difference between the treatments. But rather, the p-value is the probability that if the null hypothesis is true, then we would see a data as extreme or even more extreme than the ones we have actually saw. And if the sampling processes were to be repeated many times, we would still get the same result. So this process is still hasn't been quite answered in our original question, as we have asked if there was any genuine difference in the biomarker levels with these two treatments. So the answer is that on the assumption that there is no genuine difference in the biomarker levels with these two treatments, then our data are not particularly as extreme. So therefore, by convention, we would decide that there is no real difference between the two sampling methods. So how can this procedure lead us to astray? There are two ways that we can conclude what is wrong. The first one is that we have decided that the difference is actually real. For example, that the null hypothesis is false, but in fact, then the null hypothesis was true according to the example. In this case, this is referred to as a type 1 error. While if we have decided that there is no substantial evidence that the null hypothesis is false, but in fact this null hypothesis is actually false, then this is referred to as type 2 error. So the significance doesn't mean that it equates with the importance of the finding. And even if we correctly decide that a statistically significant difference is real, then that doesn't mean that it is important in actual presentation. This can be too small or even too to be a real of a representation to the general population, and therefore we return to this idea. So, in summary, we should choose a test of STAT, that a quantified test that can measure the effect in which we are interested in, such as the function of the difference between the mean values. And we can set up an appropriate null hypothesis, such as that there is no true difference between the mean values. And we can imagine a process of a repeatedly drawn samples of data from a population in which the null hypothesis is actually true. Or we can use a mathematical model to determine the distribution of the possible values of the test of stat if the null hypothesis was actually true. And afterward, comparing the observed values of the test of stat with this distribution, that would give us an p-value for our data. Also, some of the common statistical inference tests are defined as the derivation of the conclusion from the given information or premises. So we define the stat inference to be a derivation of the conclusion from the information in the form of data using any statistical methods and principles. So one of the most common of these stat principles is the statistical significance testing. And here we will review some of the most commonly encountered statistical significance tests, which are used to make the inference from a sample of data in any clinical research. The first type is the continuous data t-test. As t-tests are being used to make inference about the mean values in the samples of a continuous data, and we can use these tests to make an inference about the mean values in a single sample or even the differences between two mean values in a samples of data from two different groups, for example, conditions or treatment or even a patient subset. Or we can use it also to know more about the mean changes from the baseline in a paired data. So these t-tests are based on the mathematical models that assumes the underlying continuous measurement have a normal distribution. And also for conversion of the data from different groups, the simplest test that can assume that the variance in the two groups is the same. So in the case of a one-sample t-test, as in some experience, the situation we may have of a set of observation of a continuous data that were collected from a single sample of a subject or even in an observational unit. So in such situations, our interest can generally lie in making the inference about the mean value of the population from which these observations were drawn. So we can commonly ask ourselves whether the true mean has a certain pre-specified value or not, as these type of inference can be made using a one-sample t-test. An example for a one-sample t-test include this example of biomarkers that are being measured in a single sample of patients, as we have results, for example, of 12 samples as the following. Therefore, we can test the null hypothesis that the true mean is equal to 10, as we will have two just-sided p-value as an example. So using a simple program that is writing using, for example, the R analysis system, which is one of the most commonly used system to analyze any research data. So upon entering the values in the R system and choosing a t-test that is two-sided, then this will give us the following result. So the R is not a user-friendly package, but can have a useful information in the analysis process. So according to the result that we have obtained from entering the data into R, the p-value is higher than the conventional level of 0.05. Therefore, we interpret that this result would mean that these data are consistent with the null hypothesis that the true mean level is 10 and not 0. While in the case of having a paired data, as we can have a set of a continuous measurement in some cases that were recorded before an intervention is applied, such as at baseline data, for example, and then a follow-up measurement on each subject, which is the observational unit that is made after the intervention, which is called the paired data. So, for example, if we measure the participant blood pressure before starting the treatment, and then if we measured it again after, for example, 2 weeks of starting the treatment, this is called a continuous or a paired data. So in this case, we are usually interested in making the inference about the changes in the mean from the baseline to the post-intervention period. So we could quite simply subtract the baseline value from the post-interventional value, or the other way around, and then we would have a single set of differences of observations, which we could analyze as a single set of value using the test that we just described. An example of a paired data include, as we said, the numbers of subjects with their baseline measurement and their post-treatment measurement. This is an example also on the biomarker levels that we previously discussed. So if the treatment has no effect, then the true mean value of the difference between the baseline and the post-treatment is 0. And we can test this null hypothesis using the single sampling t-test that was described before, and coding this into our program. The result will be that the p-value is higher than the conventional level of 0.05. Therefore, we interpret the result to mean that these data are consistent with the null hypothesis, and these are the true median or mean difference from 0. So in the BERT analysis using R, some of the stat software packages can allow an option to perform a BERT t-test without the need to even calculate the difference between the BERT observations. This can be done in our program as the following example, and it would give us the exact same result as in the previous entering of the data. So this is a practical example of a study that measured the thyroxine level before and after an SCT. So this is how the R values will be presented in a diagram, depending, of course, on the levels of the null hypothesis. They measured the mean value and the median with the minimum and the maximum predicted values. So another common program for analyzing any research data is SPSS. It can compare the mean and the paired sample test in a similar way to the R program. Also, SPSS can conduct non-parametric tests with two related samples, such as the Wilkinson signed rank test, which is a method for analyzing any paired data. So according to the values that we just had from SPSS program and our program, it would indicate that thyroxine level post the treatment are significantly lower than in the pre-treatment condition at both 12 and 60 months of the treatment options. So this example had three types of continuous data. For the two sample t-test usage, when we have a two sample of a continuous data with the sample perhaps from different treatment groups or even different patient subsets, or just generally from two different conditions which we want to compare, then we can use a two sample t-test to make the comparison of the mean values in the two groups, or even equivalently to make an inference about the differences between the two mean values. And if there is no genuine difference between the two groups, then the true difference between the two mean is zero. Therefore, the t-test can be used to assess this null hypothesis. So an example of a two-sample t-test include measuring the biomarker values also for two groups, each of 10 subjects, giving a different intervention. So in this case, we can use a two-sample t-test to compare the mean values, as the null hypothesis is that the true mean difference is zero. So entering or coding the data into R to perform a simple two-test sample t-test as the following result, which can give us a result of the p-value that is higher than the conventional level of 0.05. So we interpret this result to mean that these data are consistent with the null hypothesis, so that the true mean difference is actually equal to zero. So to interpret these results, the time to the neutrophil engagement, for example, is 2.1 days, which has a 95% confidence interval and is considered longer for patients who are receiving a graft from an unrelated donor when compared with the civilian donor, which is given a p-value of 0.001. While another measure or method for measuring continuous data is the Wilcoxon rank sum test. So as we have noted previously, the t-test can make a number of assumptions about the data that are being analyzed. So one of which is that the underlying values have a normal distribution. And if we have reason to doubt that the validity of this assumption, then we can use the Wilcoxon rank sum test to make the comparison between the two groups of continuous measurement. So this test is an example of a non-parametric significant test, such as making a very general and widely applicable assumption about the data, and are often a good alternative to the tests that make a restrictive assumption. So in the example of the biomarkers that we have used previously, uh, of each of the 10 subjects that were given a two-drug, we can use the Wilson rank sum test to compare the data distribution and therefore their median values. So upon entering or coding the data into R and using or choosing the Wilson rank sum test, the results will conclude that these data are consistent with the null hypothesis. So this is a blotted infographic of the information that we just analyzed in our program that will be represented in a histogram of this type. While if we were using a SPSS program, this will compare the mean values and using the independent sample t-test, all of which will actually give us the same result as our program of the same data that are entered. But also using t-test is an inappropriate test for this type of data, which are a continuous data, but we should rather use the Wilson rank sum test. In the case of a non-parametric test or data, these are if the continuous data is not normally distributed or the standard deviation are very different, then a non-parametric alternative to the t-test known as the Mann-Whitney test can be utilized in this case. This is another derivation of the same test that is due to Wilson. This is the result that we will get by conducting a Mann-Whitney test, which is almost the same as the Wilson SAM rank test. So interpreting these results, it will indicate that there is a statistically significant difference in the time of the platelet engraftment for patients receiving a graft from an unrelated donor when compared with a sibling donor. So similarly, the Wilson signed rank test, which is another non-parametric test, can be used to analyze the paired continuous data in a situation in which we don't believe that the assumption of a normally distributed data is true. So we could apply this test for the example of the paired data that we saw before, and then code it in our program to perform this test. The results will indicate that the p-value is higher than the conventional level of 0.05, and we can interpret this as these data are consistent with the null hypothesis that the true median difference is 0. So, in summary, the paired versus the non-paired data can be differentiated by using different analyzing techniques. If it was an unpaired and normally distributed data with similar variance, then we should use t-test. While if it was an unpaired and non-normally distributed data or with non-similar variance or very small number, then we should use the Mann-Whitney test. While in the case of paired data, then a paired t-test or Wilson signed rank test can be used. While if we had any categorical data, which are the quantitative data that is best to be presented in a form of table, such as one variable can define the rows and another categories for the other variables, defining the column. So these data are arranged in a contingency tables, and individuals are assigned to the appropriate cell of the contingency table according to their value for the two variables. This is an example of using SPSS descriptive cross-tape for this type of continuous data. So a test for performing this type of data are the chi-square test. It is used to test whether there is an association between the row variables and also the column variables. And when the table has only two rows or two columns, then this is equivalent to the conversion of the proportions. So the chi-square test compares the observed numbers in each of the four categories and then compares them with the number that are expected if there was no difference between the distribution of the patient gender. So the greater the difference between the observed and the expected number, then the larger the value of the chi and the less it is likely that the difference is due to a chance. This is an example of a chi-square test table conducted using SPSS program. So these results indicate that the proportion of sibling of unrelated donor are different for the sibling and the female's patient from that of an unrelated donor. Another type of test is the Fisher exact test. We can use this type of test when the overall or total of the table is less than 20, or if it is between 20 and 40, with the smallest of the four expected value that is less than 5. So in this case, the Fisher exact test should be used. This is an example of mini allograft patient using the chi-square test and the Fisher test. So chi-square test can also be applied to larger tables, and it is generally called an R multiplied by C table. And when the R denotes the number of the rows in the table, while the C indicates the number of the columns. So the standard chi-square test for tables of the two rows in a general test is to assess whether there are differences among the proportions. So when the categories in the column have a natural order, where a more sensitive test is to look for the increasing or even decreasing trend in the proportion over the column. So this trend can be tested using the chi-square test for the trend, which has a 1 degree of freedom in detecting the result. So in the below table, the rationales between the frequency of, for example, cesarean section and the maternity foot size is present. So this is indicated with using three rows indications with more than 5 columns. So the standard chi-square test for this is 2 multiplied by 6, which can give us a value of 9.29. So for which the p-value will be 0.098, with also the analysis of the data for the trend that can give us 8.02. So therefore, there is a strong evidence in this case of a linear trend in the proportion of the women. So this relationship is not casual, but also reflects the size, which is a conventional indicator of the small pelvic size. So in summary, for categorical data conversion, the studies where there are two groups of the total number of patients, that is more than 40, in this case, we should use a chi-square test, where if there are two groups and the total number of patients is less than 40, or if there are more than 40 with a single cell that has less than 5, then in this case, we will use the Fisher exact test. And for studies where there are more than two groups, but they are not ordered, then we should use a chi-square test. And in studies where there are more than two groups which are ordered, then we should use the chi-square test for trends. So, for statistical significant tests, there are countless statistical significant tests that are being used. All of them produces a p-value, which is a measure of how the consistencies of the data with the null hypothesis for that test. And most of this can make some assumption about the nature of the data, and it can always assure us that we are understanding what these assumptions are, and we should always be sure that we understand that the null hypothesis is for the test we are using. And low p-values less than 0.05 can indicate data that are consistent with the null hypothesis, and a larger p-value can indicate the data that are consistent with this null hypothesis. So upon using statistical significant tests that are sparingly with care, these are not in the medical research, but they can and very often are overused and even incorrectly used with inappropriate misuse and misinterpretations of the statistical significance test and the p-value, which is very common in the medical literature. So here are some notes on the possible problems with the significant testing and also some things that are commonly done with the significant test but shouldn't be done with any stat significant test. With the multiple significant tests, one important thing is to be cautious about the overuse of any testing, and for this reason, it is important to set out in advance which conversion will be made with the significant testing. So, according to the guidelines, the stats section of the protocol should specify the hypothesis that is to be tested and the extent to which the primary analysis is planned before contributing to the degree of the confidence in the final result. So, a simple example of that, if, as in some cases, someone might use a statistical significant test with p values to compare the baseline data, such as the gender or the age. So, these are of non value to the actual result of the research, but should only be mentioned as a comparing point, not as a significant indication. So once the data from the clinical trials are available for analysis, it is therefore a common practice to carry out the test of a baseline homogeneity on a prognostic covariates before proceeding to analyzing the effect of the treatment on the outcome variables. Also, this practice is philosophically unsound and there is no practical value and potentially misleading. Therefore, instead, it is recommended that a prognostic variables to be identified in the trial plan and also fitted in an analysis covariance regardless of their baseline distribution of significance. We shouldn't also use an insignificant test result to claim that there is no genuine difference. as these tests is a method of helping to decide whether there is a genuine difference between the two treatments or not. And it is often what we want to decide when we are comparing the efficacy measurements. But in some cases, we are really hoping that there is no genuine difference between the two treatments, such as adverse events on a new drug compared with the existing standard. Therefore, a statistical significant test which gives a p-value of more than 0.05 cannot be used to support the claim that there is no genuine difference between the treatments. Therefore, a statistical significant test is considered a method for helping to decide whether there is a genuine effect that is present in the data, as opposed to the alternative, which is that the data can reflect only random and natural variability in the quantity that is being measured. And many standard stat significant tests can exist which are designed to test any specific effect, such as the differences between the means and the differences between the medians. So therefore, we should choose a significant test that is relevant and appropriate for our data and for the hypothesis that we are interested in testing. Also, a significant test which yield a p-value which tell us how the consistencies of our data are, with also the null hypothesis of the data showing any random variability only. So as we have discussed the quantitative research analysis method, now we will be discussing the qualitative methods. First of all, in a qualitative method, this includes ensuring that the research has a better quality of data. And one way of conducting this is by including the public in the research. So why do we include the patient and the public in the research process? This is considered as an ethical right, a democratic right, and a utilitarian right, and mandatory for some organizations. And also, it can help in avoiding waste research. So, public involvement can include all steps of the research process, including before we start with the research itself. This can be included in the identifying any problem and prioritizing the issues process, or even in the designing and managing the analysis methods. We can include the public with even the understanding or undertaking with the analysis and the dissemination with the implementation of the results. So, what is a qualitative research? A qualitative research needs more questions to seek the answers about a specific phenomena. That is, we don't understand much about it or we don't know any much about it, and is sometimes considered the first step before carrying out any quantitative research. So, how do we understand each issue in the world? There are different ways of knowing or understanding any particular issues, such as the positive way, which is the stable reality and the observability with the value free that is aligned to the natural science. While there is the interpretive way, which is not searching for the reality of the world, but how the people can actually interpret their world. So, by understanding what assumptions are made at the planning stage of the research, can help us in better analyzing or better implementing the research process. So, a qualitative research it is not always clearly discriminative from the quantitative research. So, this can be an example of that is laboratory data versus any social or public involvement research with an interview. And accordingly, there is always interpretation to the data. So it is not like there is an absolute answer to every one set of data, such as when we get a band of a gel or even get some kind of number. It has to do with how you set up the machines rather than you take us as a background of the control of the external variables. So all we can do in this example is interpret the things based on what we already know. So there is nothing wrong with that, but this is how the science works. So what is a qualitative research? This is a set of interpretative material practice that can make the world visible. So these practices transform the world and they return to the world into a series of representations, including the field notes, the interviews, the conversations with the photograph, or even recording of the memos to self. While qualitative researchers can study the things in their natural setting, attempting to make a sense of or even interpret the phenomena in terms of the meaning the people bring to them. So some of the key features for qualitative approaches include in terms of the research question, it helps in exploring or focusing on how and why rather than on how many. While the aim for qualitative approach is to explore, to understand, and also to interpret the result. And in terms of the data collection, it helps in a close contact between the researcher and the search participant, and it is also flexible and responsive to the context and the accounts. And in regard to the data yielded, it is rich and detailed with extensive in-content content, but also fewer cases than that of the quantitative research. While for the analysis, it allows for emergent concepts and involves the description, the classification, and the typologies with the explanations for the data rather than categorized or IDs data. And in terms of the output, it interprets the social meaning and represents the social world for the participants. So when can we use qualitative research? For example, in vaccine studies, as it needs quantitative studies to estimate the size of the target populations, the scale of vaccine production, and the service framework for providing the maximum coverage doses. While the need for a qualitative study to find out what for example, the parent or the adolescents think about this vaccine and why they believe what they do and how to overcome these challenges in order to have a better implementation for such interventions. An example of that is the FOICE trial, which is according to the epidemic of the vaginal and oral interventions as a must list of HIV pre-exposure prophylaxis trial with the target of focusing on young women in South Africa and Uganda with a daily application of 1% of gel that is daily given in an oral dose with the anterior phosphate. So the results of this trial included there is no effectiveness for the three products that were tested among up to 5,000 women at 15 different sites. This was due to the widespread product of non-used plasma level despite the high end of the study retention rate and also the high self of reported product use. Therefore, a quantitative exploratory ancillary studies was even implemented before the start of the study, and in which 100 participants from the South Africa has participated, included in focus group discussion or in-depth interview with study clinic or series of interviews. So the reasons according to that qualitative study for the non-use is the HIV stigma, the fear of being labeled as having HIV, or the ambivalence about the research process with confusion about the use of these antiretroviral therapies to prevent the infection, and also the need to balance the trial participations with other priorities and also social relationships. So, what is the difference between the qualitative and the quantitative research? As you had think about this, the different methods with different purposes can include in the quantitative research the numbers that are detected, while the qualitative research focus more on the words that are given, and it is also inductive type of research. While in the quantitative research, it tests the hypothesis, while the qualitative produce a theory. Also, the first measures the objective facts, while the second constructs the social meaning. And in the first type, it is independent of the context, while the second is a context constructed. The first type also has closed question, while the qualitative research has an open type of questions. And it can involve the researcher themselves, while the first type detach the researcher completely. And it is the quantitative rigid in design with reliability and validity and can include many cases and have some statistical analysis. While the quantitative research is considered as a responsive design with authenticity and has a fewer cases and can be analyzed using thematic analysis methods. Also, the quantitative research in function has the contextual fact of extending of the existence forms of homelessness, for example, which is can include the characteristic of homeless people, while the qualitative research can measure the nature of different forms of homelessness and experience the meaning of being homeless. So this is just an explanation of how different type of research method can analyze the same problem. And in terms of the explanatory fact, the first can explain the factors that are associated with the issue, while the second explored the events before the circumstances of the issue that developed. And for the evaluation, the first has the extent of using the service for the issue themselves, while the second has the apprised intervention experience. For the sampling technique in the different qualitative research, first of all, the sampling aim in the qualitative are the in-depth exploration and the data in the context, while the quantitative focus on the measures extent and the statistical correlation of that data. While for the sampling requirement in the quantitative, it must have a map diversity, including a key group and diminutions with a smaller sample size. And in the quantitative studies, these can include a statistical representation of the exact mirror of the population with larger sample size and of the estimations. So in the first one, the sample type is purposive as a deliberative selection on the basis of knowing the characteristic factors, while the second, the quantitative, have a random probability of the sampling to ensure appropriate representations. So there are four sampling techniques in the quantitative research. This is the purposive, the theoretical, the convenience, and the snowballing methods. So the sample that is selected with the purpose to represent a type in relation to the key criteria that is suspected to be relevant. This can include the socio-demographic, the experience, and behavior roles, with two aims to ensure that all relevant key criteria are being represented. This can also include the diversity within each criteria and with different approaches, such as the homogeneous or the heterogeneous, with the extreme cases and the key information. In the theoretical sampling, the sample based on their potential contribution to the development and also the testing of theoretical constructs. It is also associated with the development of a grounded theory on the initial sample selected and the data that are analyzed with further samples that are selected to refine the emerging categories and their theories. So this is useful in the exploratory research where there is little knowledge about a particular phenomenon. While in the snowballing and convenience sampling, these are series of introductions to a new sample from research participants, and it is considered useful for the harder to reach and easy to ignore groups. And in terms of convenience, these are chosen or according to the ease of the access for them, but it can lack the clear sampling strategy. In regard to the method for inquiring in qualitative research, these can include observation, which can be combined with participation, and also archives or any other type of documentary sources, including media. While interviews can be also semi-structured or even narrative, and one-to-one interviews or with even focus group that are present. If an interview was chosen as the method for collecting the data, there are also different types that are to be constructed. The first is the structured interview, which are used in the quantitative surveys. These are a set of standardized questions, fixed wording, or any other order of questioning with a predetermined categories, which are part of a large sampling survey. While in the semi-structured interviews, these are used in the qualitative studies, and it is somewhat more flexible with a standardized interview schedule with a topic guide, and it have exploration of the emergent themes and ideas with limited dissertation or even disclosure. And in the unstructured interviews, these can be used also in the qualitative studies as the style is unstructured with only some gentle guidance and has a biographical stories with narrative. So if an interview was chosen, there are some topic guides that can help in aiding the interview. This help with having a flexible guide with document scope and also coverage of the interview, which can also include the key sections with the key subtopic and ordering of the topics that begin with the easier and less sensitive types of questions that has also a positive and polite interview procedures with topic guidance. And if we were using questions for depth and breadth, as for example, discussion with the questions for breadth and depth, these should be a short open question while avoiding leading questions or double questions, such as, could you repeat that answer for me, which can lead the interviewer or the person being interviewed to change their answer. Also, the content mapping to raise an issue or the content minding to explore any issues. So, when conducting an interview, this will help in having rapport and trust with the participant. We also must be natural and reassuring in using the approaches, and we should avoid any self disclosure, and we should allow the representative time to think about their answer, but also listening carefully to them with a role as a researcher, not as a counselor or an advisor or even a healthcare professional. This should be audio recorded with matching the interviewers and the interviewees, depending, of course, on the type of the study. Where if a focus group was being used, this can include 6 to 8 participants with interaction between the participants themselves and composition of the carefully selected participant. This can help in encouraging the exchange of the views and the experiences. So, what are the differences between the in depth interview and the focus group? In the in depth interviews, they can include even personal stories with detailed or complex issues and even sensitive issues. While focus group can be generated by group interaction, a broader understanding and not too sensitive content in regard of the questions that are being answered. We will be taking a break now and then we'll continue with discussing the qualitative data analysis.